For those of you who don't know me, my name is Evangeline Alosilha, and it's my privilege and great pleasure to welcome you to the East and Southeast Asia session. On behalf of GARD, I want to thank the speakers and the regional leadership team. GARD's vision is to prevent illness, save lives, and sustain health through rapid diagnostics, especially for populations who need help the most, but can afford the least. Sustainable health includes the human body and everything else that maintains it: safe and nutritious food, clean water, and a clean environment. Whatever your field of interest, you have a place in GARD, and we welcome you to join us. Now I want to introduce our speaker for the opening remarks, Dr. Siddharth Chandra. Dr. Chandra is the director of the Asian Studies Center at Michigan State University, and he is also a professor of economics at James Madison College at Michigan State University. Now I will open his video for his opening remarks.
Good afternoon and good evening. My name is Siddharth Chandra, and I'm a professor at Michigan State University and director of the Asian Studies Center. I'm delighted to welcome you to the East and Southeast Asia session at the 2023 GARD Forum. As you probably know, there are over 500 registered participants in the forum from 28 countries on 6 continents. There are 8 regional technical sessions from East and Southeast Asia, South Asia, Africa, Europe, Oceania, Latin America, North America, and Asia, Africa, some of which have already occurred. We have 90 speakers in various sessions, 15 short courses, more than 50 teams competing in the Innovation Challenge with prizes, 6 women scientists from around the world speaking at the GARD Women in STEM session, and a special roundtable featuring Borlaug and Scientific Exchange Fellows. Over the years that the Asian Studies Center has supported Professor Evangeline, our dear colleague, Professor Evangeline Alosilha's efforts to build the GARD Forum and the GARD Alliance, we are delighted to see all the progress that has been made. We have seen a number of very innovative programs held prior to this session as well. These include efforts to take technologies that are being developed in the area of rapid diagnostics to market, to try to infuse these efforts with innovative ideas from around the world. And we believe that the end results of these efforts are going to have enormous impacts globally. I cannot overemphasize the importance of rapid diagnostics that are also cost-effective, reaching the far corners of the world. As you probably know, the cost in terms of human lives and misery of easily diagnosable and preventable diseases is enormous. And so it's with great pleasure, but also deep humility, that I welcome you to this session. And I hope that it's a successful one in building networks of like-minded colleagues to continue this wonderful research and to bring some of your ideas to fruition to prevent disease, save lives, and increase the levels of happiness worldwide. Thank you.
So with that, I give the floor to Taz or to Paula.
Okay, it's going to be me, ma'am. Thank you very much, Dr. Alucilia, and also to Dr. Siddharth Chandra for the opening remarks. And of course, I welcome everyone right now, to all of our participants who are watching right now for the East and Southeast Asia region of GARD with a seminar or webinar title, Innovative Rapid Diagnostics in the One Health approach perspective. So I am your Master of Ceremonies, Dr. Tadeus M. Carvajal, an Associate Professor in De La Salle University. We have today invited 4 speakers who are actually experts and who have experience, okay, in using rapid diagnostics in both in the One Health perspective. So first, let me first introduce to you our first speaker. Okay, our first speaker is. Okay, I'm sorry for about that. Okay. Okay. Our first speaker holds a PhD in veterinary medicine and is a professor at the Center for Southeast Asian Studies in Kyoto University. He is a veterinarian and microbiologist. He specializes in food microbiology and infectious diseases for both humans and animals, and has been particularly involved in the development of LAMP and other molecular-based testing methods, as well as POCT or point-of-care technology for fieldwork for over 15 years. Currently, he is collaborating with local researchers in the Philippines and in Vietnam for the control of transboundary animal diseases, or TAD. Without further ado, I would like to call Dr. Wataru Yamazaki with the title Development and Clinical Evaluation of a Highly Sensitive Point-of-Care Testing for Early Diagnosis and Early Containment of African Swine Fever. So let's give a virtual warm of applause to Dr. Wataru Yamazaki. Sensei, you may share your screen.
Okay, give me 1 minute. I'll share my screen. Okay, can you see my screen?
Yes, Sensei, we can see it.
Okay, okay. Thank you very much for the introduction, Dr. Carvajal. Let's go to my session. My title is Development and Clinical Evaluation of a Highly Sensitive POCT, or Point of Care Testing for Early Diagnosis and Early Containment of African Swine Fever. First of all, thank you for this invitation to be given this opportunity. Before going to my session, I'd like to briefly introduce my affiliation, Center for Southeast Asian Studies, CIAS, Kyoto University in Japan. The Center for Southeast Asian Studies was established in Kyoto University, Japan in 1963. It is a very unique institute, first institute in Japan to conduct comprehensive research on Southeast Asia. The research we are conducting research that integrate the humanities and science, not only natural science. We are aimed for integrating the humanity and social science and natural science. It is a very unique institute in Japan. This is also a brief introduction of myself. My specialized field is food hygiene and infectious disease. My research interest is development of pathogen detection technology and international networking in food hygiene and infectious disease control, both for humans and animals. So it is my great honor to join this meeting. I hope expand my networking on food hygiene and infectious disease control via this presentation. This is today's topic. Today's topic consists of 3 parts. First one is importance of early detection technology. Second one is principle of LAMP method. And finally, development and clinical evaluation of a highly sensitive POCT for the diagnosis of African swine fever. Let's go to the first topic, importance of early detection technologies. As you know well, not only COVID-19 or African swine fever, so many infectious diseases have newly emerged and re-emerged in our history. For example, smallpox, cholera, plague, influenza, etc. I have one question. who will pay the price of a pandemic? Answer is very easy. Of course, we have to pay the cost. We have to pay it. This graph showing the relationship, emergence of a pandemic or outbreak in our history. Horizontal axis is the years from 1900 to current. Vertical axis is the number of deaths, and the size of the circle is the economic growth. This means the COVID-19. Also, this is the Spanish flu occurred in 100 years ago. The difference of color is how the virus spread to the continent. This color means the spread to the 6 countries. That means a pandemic. And the other is spread to 3 or 4 continents, or fortune case was only 1 continent. This is the SARS-1. This is influenza H1N1. This is Nipah. This is Ebola. So anyway, globalization in recent years has clearly accelerated the emergence and spread of new pathogens. And the economic damage is growing year by year. Eurasia continent account for 80% of global pig production. Asia account for 56%. Among these, as much as 50% of these pigs, or half of the world pig, are produced in China. Also, the economic damage is over $141 billion in China as of 2019 by the spread of the African swine fever. This damage is now even greater. If it occurs in the United States and early containment fails, the economic loss is estimated around $50 billion. So this is one of the reasons early detection and early containment is essential to minimize the economic damage of the pandemic or outbreak. This is the view of the damage caused by the African swine fever in Russian Federation in 2017. To prevent the spread of the virus, so many pigs are slaughtered. As you know well, currently ASF, African swine fever, has turned into pandemic. In the 2000s, this infection, ASF, spread from East Africa to Southern Europe. It slowly spread throughout Europe and the Russian Federation by wild boars. It was tragic that it spread to China in 2018. This is because China has the largest number of pigs in the world. In other words, China has become the epicenter of ASF, then spread to Southeast Asia. Also, in 2021, it finally spread to Republic Dominica in the Caribbean country, and there are fears that will be spread to the Americas. The epidemic of ASFV caused a major decline in pork production in China and a dramatic increase in imports, with major socio-economic impact. That means after spread of the ASF in China, pork production decreased drastically. In contrast, imports of the pork product increased, skyrocketing. Why did it spread from Russian Federation to China? The researchers suspected wild boar, but that was not the case. The miserable pigs are victims of political performance. Please recall that in 2018, the Trump administration in the United States imposed a high rate of sanction on China. China found it difficult to import cheap US pork products. Instead, in China, imports and potentially smuggling from Russian Federation and Eastern Europe increased dramatically instead. Unfortunately, these meat products from Russian Federation or Eastern Europe were contaminated with the ASFV. In summary, in 2018, the US Trump administration imposed high sanctions tariffs on China. China has had difficulty importing inexpensive US pork and pork feet. A cheap US pork product had been no longer exported to China. So instead, in China, pork product imports and smuggling from Russian Federation and Eastern Europe increased. A pork product from Russia and Eastern Europe was contaminated with ASFV. This is the reason why I speculated. The ASF epidemic in China 2018 spread from coastal area with ports, such as Shanghai, Shenyang, etc., not inland, not from Mongolia, not from Russian Federation, not because of wild boars, but because ASFV entered the country from pork product, landed at the port. This is a speculation based on the epidemiological data. Okay. Hence, I would like to point out the importance of minimizing the damage through early detection and early countermeasures. Early detection and early countermeasures can reduce the damage of animals. This graph shows the light green indicates the damage caused by countermeasures taken. Dark green indicates damage caused by countermeasures not taken. That meaning early detection and early countermeasure is very important to minimize the economic damage or spread of the infectious diseases. Also, this is not today's main topic. It can also potentially reduce the risk of cross-species transmission or spillover from animals to humans. As in the case of COVID-19, a red indicate when countermeasures are taken, and light blue indicate when countermeasures are taken. Both of the case, if we can successfully take a countermeasures early phase, it can be possible to minimize the damage or spread of the diseases. But if failed, damage is spread rapidly. Okay, let's go to the second topic, principle of LAMP method. LAMP, or loop-mediated isothermal amplification, is an established nucleic acid amplification method developed in Japan in 2000, offering rapid, simple, and cost-effective diagnosis of infectious disease. This is based on the unique principle depend on 6 specific primers recognizing 8 regions, and the use of chain replacement enzyme make it possible isothermal amplification. Also, LAMP allows one-step detection of gene amplification at a single temperatures, such as 63 degrees or 65 degrees, and requires only a simple incubator, such as heat block or water bath, providing a constant temperature. Also, judgment of LAMP is based on the naked eye analysis, such as the color change of the reagent. So expensive detector is not required. Also, we are using CFI. CFI is color fluorescent indicator. This is firstly developed by the Japanese researcher, Dr. Hayashida, in 2015. HNB or hydroxyl blue and gel green are used for color change and fluorescent judgment for improved lamp reaction. Color change is confirmed by naked eye. Initial reagent color is purple, but the positive turn sky blue. Also, fluorescent detection is possible. Fluorescent is confirmed in real time by qPCR machine using film filter or portable illuminator like this. Okay, for our better understanding, let's watch the animation video explaining the lamp principle. Because all Japanese like animation, so let's share this. Give me 1 minute. I'll shift the screen, sharing the screen. So, これね。Can you see the animation screen?
Yes, Sensei, we can see it.
Yes.
Okay, thank you. Thank you. I'll start the animation video. This animation explaining the lamp principle. This pale blue is the enzyme. It works at the isothermal temperature such as 63 or 65 degrees. Like this, this is a dumbbell-like structure. It's base of the lamp amplification. Primers anneal this structure automatically, and the amplification also accelerated at the isothermal temperature. Apart from, it is different from real-time PCR. PCR requires temperature change from 95 to 60 and again to 60 to 95. It is time-consuming. However, in contrast, lamp requires isothermal temperature, no need for temperature change. So, as a result of difference of the principle, lamp make it possible the early detection, early amplification. Size of the specific amplified product is various. However, each of the product is specific. So it enables the rapid detection. Again, for our better understanding, let's repeat the watching the video. Okay, thank you for watching. I'd like to go back to my presentation file. To give me 1 minute, I set up my computer. I'll share my screen again. Can you see my screen?
Yes.
Okay, thank
Yes.
you. Okay, thank you very much. Thank you for sharing the animation video. As I explained, LAMP is very suitable for cost-effective diagnosis especially in the field, because the expensive detector is not required. So finally, I'd like to go to the last topic, the development and clinical evaluation of a highly sensitive POCT. This is based on LAMP method and for the diagnosis of African swine fever. We have developed a highly sensitive POCT, or point of care testing, that can be performed using pig whole blood samples collected on the farm with only simple equipment. This is a protocol, DNA extraction, and then LAMP amplification. In total, 20 minutes to less than 1 hour is required for the beginning of the detection and the final judgment. And this is the point of DNA extraction. Usually we use a commercially extraction kit. It is tedious and time consuming. Usually around 30 minutes will be required for DNA extraction. However, we use SDBS. This is a special reagent, just mixing the whole blood and SDBS 1:1 ratio. Then heat at 95 degrees for 5 minutes at heat block and centrifuge for 5 minutes. We are using the portable centrifuge. However, usual centrifuge is also acceptable. At the full speed, around 1000 G to 2000 G when we use laptop centrifuge, but it is enough. A supernatant containing crude DNA can be used as a DNA template. This is very dirty color. However, in spite of this dirtiness, it can be possible to detect by using LAMP. This is a LAMP detector, real-time detector. If we can use it, it's much faster. We can judge the result real time. However, if it's not available, instead we can use heat block, simple heat block. This makes it possible endpoint analysis. In this case, we use portable detector or naked eye observation. Anyway, in this case, we usually use 45-minute amplification. So, in total, it is very simple and cost-effective detection system, I think. Total reagent cost per one sample is less than $1. It is very cost-effective. This is the performance of the POCT from whole blood compared to conventional method. Conventional method is the kit-based column kit DNA extraction from whole blood and real-time PCR amplification. This is carried out in the laboratory. Compared to the conventional method, comprising of DNA extraction kit and real-time PCR, our developed POCT showed 100% concordance, demonstrating it is extremely high performance. When we used 89 whole blood samples, diagnostic sensitivity and diagnostic specificity are perfectly matched. In Vietnam, the clinical evaluation was conducted with pig whole blood samples collected from ASF outbreak farms in north part of Vietnam between 2020 and 2022. The result is summarized in the paper and now is in press. Result can be judged like this. Read out by the naked eye. This is positive samples. Negative sample is red. This is the pigment of the whole blood. So positive they turn into dark green, but it's possible to differentiate, but sometimes may be difficult. So in this case, we can use LED, a handy LED. This is much clearer. Easily differentiate the positive from negative. Also, we have carried out the comparison of LOD. LOD, the limit of detection, was compared using 10-fold serial dilution. Positive sample is diluted by using the negative pig whole blood. As a result, the developed POCT was 10 times less sensitive than conventional method. That means this is the limit of detection. A conventional method turned positive until 10 to 5 dilution, but 10 to 6 negative. In contrast, our POCT turned positive until 10 to 4, but 10 to 5 showed negative. So 10 times less sensitive, our developed method. However, the high viral load in the clinical whole blood sample, it is very high, so which did not cause false negative in the clinical evaluation in Vietnam. This is the example when we use the extracted DNA, not crude DNA, but the purified by using the column kit extraction. In this case, it's very clear. Negative shows the violet. However, a positive sample turns blue. It is very clear. However, this is the when we use a crude DNA from whole blood by using SDBS. The color is not clear. This is the color pigment of the whole blood. So blue plus red turns green. So this is the positive sample show green. Maybe it is, sometimes this is difficult to distinguish by naked eye, but when we use LED, it is very clear to distinguish. So in summary, we successfully developed the POCT, highly cost-effective and relatively sensitive. That's all my presentation. Thank you very much for your attention. Salamat sa iyang attention. Thank you.
Okay, thank you very much, Yamazaki Sensei. Okay, of course, thank you for providing us some basic info about LAMP technology. I think majority of our participants who are watching now would appreciate this particular technology in trying to do a rapid detection kit, especially for African swine fever. Now, Dr. Yamazaki Sensei, there's a question here by one of our participants, and he said that, may I ask just about the most common technical difficulties that we might encounter when developing and utilizing LAMP technology? So meaning, Sensei, that probably what could be some potential limitations about about using LAMP technology?
Thank you for your question. One of the bottleneck of the development of LAMP is the difficulty of primer design. Usually, PCR use only 2 targets, or in case of the qPCR, only 3 targets is. However, LAMP is required 8 targets. That means comprising of 6 primers. That means a highly conserved region, around 200 base or more, is required. Initially, we have to find the highly conserved region, more than 200 bases. This is the point. It is make it difficult, the LAMP development. In the case of ASF, this is DNA virus. So also it is relatively easy to find the conserved region. But in contrast, such as influenza or norovirus, this is RNA virus, and with very high variability of the sequence. In these cases, it is very difficult to find the conserved region. In this case, usually we give up to design the LAMP primers. So it depends on the limitation is this is a very weak point and the limitation of the LAMP. So, but once we find the conserved region, it's relatively easy for DNA virus or bacterial pathogen. In this case, it is very suitable. However, RNA virus, it is sometimes it is difficult to find the conserved region, make it difficult to develop the LAMP. I hope this meets your question.
I think, Sensei, that is indeed correct. Of course, based on my experience, just basic information, that LAMP actually requires a lot of your, like, primers, if I call it that way, in order for you to amplify that region. But maybe, Sensei, what could be the best way in order for someone to hurdle this? I know that there are, if you are doing a somehow first time, like a novel technology for or a novel LAMP technology for a pathogen, what would be your best advice on developing those primers or the primers needed for LAMP?
Sorry, to be honest, I don't fully understand what you
Okay.
mean.
I mean,
could you summarize the
yes.
point?
All right, Sensei. So I know, Sensei, you require a lot of what you call primers, right? In PCR, you would just need only two, right? The forward and the reverse. But in LAMP, you said you need at least 8 for that particular region. Am I correct on this? Yes. So what would be the best way to identify the 8, Sensei? Would you need somehow first a very long sequence in order for you to identify the 8 regions or the 8 for LAMP in this particular case, Sensei?
Thank you for your question. Usually, by using the data of the gene bank, we retrieve the target sequence and we carry out the alignment, then find the highly conserved region. So once it find conserved region should be high GC content. If the sequence contains a lot of A or T, it is not suitable because it is impossible to maintain the high temperature for the reaction. So, once we find the conserved sequence with high ratio of the G and C, then we can automatically synthesize the specific LAMP primers. Then, it is difficult to explain, but if
yes.
once we find the sequences, we trial error. So automatically design the LAMP primers 3 or 5 sets. And if I'm not sure which is the best sequence, but we carry out the preliminary trial on the primer set showing the high performance, high speed. We can adopt it, and then we switch to the clinical evaluation.
And in your experience, Sensei, there's a question here. How long does the color last in the LAMP technology before degradation? Would you happen to know how long that particular color would actually degrade over time?
Say that again. You asked about
sorry.
amplification temperature.
No, Sensei. Remember, Sensei, that you try to have like the CFI, or I'm sorry about that, the color, the fluorescence in the LAMP technology, because you have to put UV in order for you to know whether it will be positive or negative. So the color that you see, does it degrade over time? For example, that if you are able to have like your target DNA, is there a limitation on the time for the color to stay at S or fluorescence?
So sorry, if you can use the chat box for your question, it is very
Yes. Yes,
Is
I
what
think,
you mean?
yeah. Yes, Sensei. In the chat box, you can see that in the chat box, you can see the question
Yes,
of Miss Budu. Okay.
Miss Voodoo. Okay, I can see it. Wonderful. How long does the color last? Okay, I got it. okay, thank you. I have correctly measured the how long it is stable, but actually around 30 to 60 minutes will be enough, stable enough. Because after amplification, we use the inactivation step at 95 degrees for 2 minutes or 80 degrees for 5 minutes. This inactivation step make the reaction stable. So we can confirm the color change stably more than 1 hour or more. Even though overnight we checked the result next day, it was also stable. So
Okay.
we have never confirmed the degradation. It was very stable.
Okay, Sensei. All right. And there are other questions here, Sensei, in the chat box. Maybe we can just entertain one more. Okay. Let me just check. So there's a question by Dr. Kwang. Okay. KP Kwang. Okay, Dr. KP Kwang.
Combine the DNA extraction step and ramp function step into one step. Two, when the reagent was patented by the ramp inventor and the very action. Okay, how to reduce the cost of the. Okay, I have just read out the 2 questions from Dr. Kwan. First question, combine the DNA extraction step and ramp function. To the best of my knowledge, seamless reaction is impossible because first and second step should be separated. Because first step we use 95-degree amplification. This temperature is enough to inactivate the polymerase for ramp reaction. So once we carry out 95 degree for 5 minutes procedure to detect DNA, dissolve the cell wall of the blood cells. It is essential. Then we have to keep the room temperature. Then supernatant, after obtaining supernatant, should be added the lamp mixture. If we do it simultaneously, BST polymerase, or essential for lamp reaction, it will be inactivated at 95 degrees. Or if we carry out this, that means direct detection of the whole blood into the lamp reagent. And then if we omit the 95-degree amplification, it was impossible to detect DNA or inactivate the potential inhibitors from whole blood. So this step should be essential. These 2 steps are essential. To the best of my knowledge, it is impossible to emerge as a one-step reaction. I'd like to go to second question. Actually, as Dr. Kwan mentioned, commercially lamp kit is very expensive. However, patent is not has been already expired, I think, because the manufacturer is Aiken Chemical Agency. Obtained the patent around 2000 or before. So patent right is around 20 years. So this is expired. Also, research use only. It is not for research use. It is not essential to use the commercial kit. A component of the lamp reagent is in public. So we usually use homemade reagent. That means we made the lamp reagent from the scratch. This is very inexpensive. In this case, homemade reagent and designed homemade primers. In total, less than $1. So if you use the commercial kit, as you mentioned, it is expensive. However, now we can use for research use, it is free from patent. Also, for the clinical use, I think it is now the patent is already expired, I think. Hope this meets his question.
Yes, thank you. Thank you for your response. Okay, answer.
Thanks.
All right. Okay. Thank you very much, Dr. Kwang, for that. And of course, I think that will be our last question, Wataru Sensei. Okay, so I know that there are a lot of questions still, but you may contact Wataru Sensei regarding the LAMP technology for African swine fever. Now, to present actually the certificate. Okay, so next slide, please. Okay. Okay, so of course, the Global Alliance for Rapid Diagnostics in Michigan State University presents this certificate of appreciation to Dr. Wataru Yamazaki of the Center for Southeast Asian Studies, Kyoto University, for the presentation entitled Development and Clinical Evaluation of a Highly Sensitive Point-of-Care Testing for Early Diagnosis and Early Containment of African Swine Fever at the 2023 Global Alliance for Rapid Diagnostics Forum with the team, the role of rapid and innovative diagnostics in the One Health perspective, held last March 25, 2023, signed by our very own founder of the Global Alliance for Rapid Diagnostics, as well as Program Director of the Nanobiosensors Lab and Professor of the Department of Biosystems and Agricultural Engineering of Michigan State University, Dr. Evangeline Alucilia. Okay, and signed also yours truly, okay, Dr. Tadeus Carval. So let's give a virtual warm of applause to Dr. Wataru Yamazaki for his presentation. Thank you, Sensei, for your time. It's very informative, and we're hoping that a lot of our young scientists in the East and Southeast Asia region would actually use this technology not only for African swine fever but also to other foodborne pathogens, as well as other waterborne pathogens as well. Thank you. Arigato gozaimasu, Wataru Sensei.
Thank you very much. Thank you for everyone. Thank you.
Okay.
Bye-bye.
Okay. All right. So thank you, Wataru Sensei. So we will now move to our second speaker on this case. So let me just try to get my notes here. So our next speaker is somehow I would consider the a scientist in the Philippines that we all look up. Okay, so parang we call it our idol. Okay, so he is actually a molecular microbiologist and an infectious disease specialist. He took his bachelor's degree in microbiology at the University of Santo Tomas and his medical degree and specialty of in internal medicine at De La Salle Health Sciences Institute in Cavite. He went to the University of the Philippines for his subspecialty training in infectious diseases. He received a research scholarship to the International Training and Fellowship on Emerging Infectious Diseases at the Center for Global Health. Global Health at the University of Virginia School of Medicine. He also holds a master's degree in business administration from Ateneo Graduate School of Business. He is a diplomate and fellow in infectious diseases. He joined the National Institute of Health Philippines and became director of this of the National Institute of Molecular Biology and Biotechnology. He dedicated his work to the identification of diagnostic targets and evaluation of diagnostic platforms best suited in developing country scenario. His most important work is the Lab in a Mug project, which functions as a multi-infectious disease diagnostic platform with its principal product, the Biotech M Dengue Aqua Kit, that received local and international recognition, such as the NSTW Outstanding Technology Commercialization Award, given by the National Academy of Science and Technology in 2016, as a gold medalist in the 46th International Invention Exhibition in Geneva, Switzerland in 2018. Gawad Dagisek for Medical Sciences, given by the Department of Science and Technology in 2019. Uh, Through the Technology Transfer Act of 2009, he built the Manila Health Tech Incorporated, the very first university spin-off company from the University of the Philippines in the area of health biotechnology. He is a recipient of the national and international awards, including the Bill and Melinda Gates Travel Scholarship in Molecular Helmintology in 2005, Outstanding Young Scientist of the Philippines for Medicine in 2008, UP Manila Research Productivity Award, the top 20 Young Physicians Leader from the Inter Academy Medical Panel in Berlin, Germany, and the Outstanding Young Men, or TOIM, all given in 2011. Dr. Jose P. Rizal National Award in Research from the Philippine Medical Association, and Dr. Eusebio Y. Garcia Award in Molecular Biology and Molecular Pathology from the National Research Council of the Philippines in 2015. In 2019, he received the Presidential Lingkod Bayan Award from the President of the Philippines, and in 2020, he was named among the 100 Asian scientists in the Asia Science magazine. His latest works is the local RT-PCR kit for the detection of the SARS-CoV-2 virus. Okay. And of course, without further ado, may I call on Dr. Raul V. Destura to give us a presentation entitled The Molecular Diagnosis for Infectious Disease Program of the Philippines, Challenges and Lessons Learned over the Last 15 Years and Future Opportunities for Growth. So let's give a virtual warm of applause to Dr. Destura. And of course, sir, you have the floor already. Thank you very much.
Thank you, Dr. Carbajal, for a very, very generous introduction. Let me share my slides. So I will be talking about our journey in the molecular diagnosis program here in the Philippines. It's all started several years back when I returned from my research work in the US at the University of Virginia School of Medicine, and have joined the National Institutes of Health in developing programs for the country. My disclosure, as already mentioned, I'm the founder and chief executive officer of the Manila Health Tech Incorporated group of companies, which is a biotechnology company focused mainly on communicable and non-communicable diagnostic platform development, including artificial intelligence. So our principal focus is really on the development of technologies that can be accessible. One of the challenges in developing diagnostics is really access. Sometimes the diagnostic technology is really, really good, but in terms of accessibility through price or costing or equipment requirement or infrastructure needs in order to run those technologies makes the technology less accessible than it should be. And then you add into the picture the landscape at which almost everything is out of pocket. So this is where the idea that we have thought about. But I would like what I always tell my mentees and my students, An idea will just remain an idea unless you execute them. So today I'll just give you an overview of the work we've had over the last 15 to 16 years, and that where the idea went, what happened to it, and where it's actually heading. In developing diagnostic platforms here in the Philippines, one thing that we have realized is even if you love certain platforms, you love certain technologies, you cannot just go dive in and start developing one. You have to go out there and look for what the problem is, especially if your resources are limited. So needs assessment is very crucial in designing technology platforms for specific targets in your own location. So basically, you identify needs that are actually needed, and that's where you're heading. So the cost-limiting nature of the molecular tools for diagnosing infectious disease in the Philippines led to the creation of the Molecular Diagnostic Program while I was still the director of the National Institute of Molecular Biology and Biotechnology way back in 2007. We created several diagnostic platforms spanning from vector-borne diseases, tuberculosis, multi-drug resistant organism, HIV drug resistance profiling, influenza, neglected tropical diseases, and waterborne parasitosis. This program has been adopted as the first flagship program for communicable disease of the Philippine Genome Center and NIMPB, UPNIH in 2009, which created the Labinamag projects, which I actually mentioned. It is an isothermal amplifications that identifies dengue of all 4 serotypes and has actually been, I think it was mentioned by Professor, our previous speaker, of an isothermal amplifications, which we converted into a closed system that allows you to visually read the results. It passed evaluation with the ASEAN Novel Diagnostic Innovation Panel in 2019, and as mentioned, bags gold medal at the International Invention Exhibition in Switzerland in 2018. But the problem is, yes, we have a technology that is multi-awarded, but then the adaptation of the technology is a little bit of a challenge. You create one, but then how are you going to bring such technology all the way down to the target end user? In the landscape in the Philippines, we're actually working on technology platforms. At that time, there were no company willing to take a chance because we are virtually untested. There were no expertise in diagnostic kit manufacturing in the country. So how can you scale those up? There was limited knowledge and technology commercialization from bench to bedside, which is actually a very challenging environment for most virtually all technology developer. So we were deciding, will it be a spin-off, a startup, or a spin-out? But since we don't have the resources and we wanted the support of the university as well as the support of the industry, we decided to go for a spin-off technology. So what happened to that particular idea? We took advantage of the Tech Transfer Act of 2009 that allows the commercialization of researchers within the university to create, own, control, and manage company or spin-off for commercialization without the need of actually severing your ties from the university, because my principal passion is still teaching. And in March 31 of 2016, the licensing agreement of the university and the spin-off company was actually approved by the Board of Regents of the university, giving rise to the birth of the first university spin-off company of the University of the Philippines, Manila Health Tech Incorporated, which has 4 major pillars in diagnostic services, isothermal solutions, PCR technology, and avian-derived technology. But creating a spin-off is one thing, running it is another. So there were a lot of challenges along the way because the regulatory mechanisms and the rules related to local spin-off is a little bit, it's relatively new. So we became the guinea pig in every steps of the way. So it was a very challenging journey for the first spin-off, and we have to recalibrate our approaches. When we were spinning off the dengue diagnostic technology, and then COVID came. 2020 became our tipping point. And because of the COVID-19, the world has become exposed on the importance and the value of diagnostic biotechnology discipline. I remember sending this viral message to my chief science officer that there is an ongoing outbreak in Wuhan, and there is a chance that it might enter our country. And we have to prepare ourselves similarly when we created the H1N1. So in 2019 of December, you all know that the novel virus that were identified around the world spread like wildfire. And the Philippines, similarly with other countries, were cut off from the global lockdowns, affecting not only movement of global goods and services, as well as access to diagnostic technologies. And as the world scrambled for the ability to be able to test individuals, so as the limitations to access diagnostic technology, which was recommended the RT-PCR. So the solutions that we can only think of is to create the response to the supply and demand, because at that time the cost of the testing was so high, from 80 to 200 US$ in the Philippines because of the varying landed cost. And the only way to make it accessible is by local innovation. So we created the Gen Amplify RT-PCR technology that simultaneously detects 3 genes of interest, 2 from the virus, and 1 as a human endogenous control. But the technology is not just about developing it, but what actually how it actually enhanced the capability for a manufacturing platform here in the Philippines, from manual to fully automated and robotic manufacturing facility, generating 20,000 COVID tests per day as manufacturing output. But the challenges was almost insurmountable. Resources was an issue. We have problems with supply chain, lockdown restrictions, too many bureaucracies, and even security. But despite all that, we were given a chance by local adaptation, 0.3% chance compared to foreign technologies entering the market. But this 0.3% actually generated over 140 jobs for security guards, receptionists, messengers, encoders, medical technologists, and young scientists. It financed the development of 57 other diagnostic technologies and medical devices, which led to the release of new products for both human and animal health in the market. So these are the platforms we have put together for both animal and human health. And recently we got an FDA approval for the Gen Amplify African swine fever PCR detection kits that has been field validated by the Bureau of Animal Industry. The other one are for neglected tropical diseases that detects salmonellosis, leptospirosis, and schistosomiasis, converting the open lamp system into a closed tube system by integrating Quasar probe-enhanced isothermal amplification technology, which received the 2022 GAWA Dagistic Award by the Technology Commercialization Unit of the TAPI Department of Science and Technology. And because of that, we wanted to make sure that technologies can actually be brought all the way to the end. We created the laboratory project that allows us to bring PCR and isothermal amplification technologies all the way to the field without actually building a 13 million peso molecular laboratory facility. So this particular platform, we partner with industrial designers and molecular biologists to put together this unit that allows them to be brought in a trolley suitcase and have your mini station laboratory anywhere you are for both human and animal health nucleic acid amplification technology platforms. So it has been tested by clinical microbiologists and medical technologists to make it really, to determine its usability. You can plug it in any AC detection port anywhere you are to run a full molecular laboratory. But the missing gap here is very high, despite of all of this experience over the last 16 years. Majority of our activity is in research and development to generate new knowledge and potential technologies that could enhance human lives. In most cases, it ends with the publication in mind, wherein it's a requirement for us to stay in the university. It's a requirement for promotions. But because there are so many gaps to encourage researchers to go to translational aspect of technology development, principally because of the lack of awareness of existing mechanisms for knowledge translation, the processes are too rigid and lack of standardization and efficiency. There is no ready infrastructure that will assist startups maximize their capital on production and scale up and not on infrastructure cost. So there is, based on what I have seen so far, I strongly encourages those who are in a diagnostic platform to push your technology all the way to technology transmission by being brave enough to study what startups or spin-offs are in terms of creating these entities. No, in this particular side of the spectrum, publications and patent applications become the starting point, and technology commercialization, scaling up, and distribution to the primary target end user becomes the end goal. Most researchers and science innovators are unaware of existing mechanisms that will support knowledge translation to innovative products and services in terms of laws that are available in the country. So these are some of those with some revisions in the Magna Carta of the RA 8439, removing the caps allotted for science and engineers. The infrastructure that we should all hopefully work together to convince our government that from an RDI, R&D in universities and other RDIs, we certainly have a lot of this already going on. And to further capacitate and train and enhance the capability of our tech transfer and business development offices in our universities and intellectual property management, to creating an environment wherein there are real, I say real because we have existing business incubator facilities, but most of them are actually occupied by businesses like call centers and all, but not really the technology business development sides. We need CGMP grade facilities for drugs and vaccines, CGLP grade facilities for biologics, synthetic peptides, cellular therapeutics, and business innovation management system. And this is where what we should be building. Things that I'm telling all my colleagues everywhere I go, every chance I get to speak, and things that I'm also telling myself to do. Why the need for the partnerships? Because of the dual R&D process. In the academy, we do fundamental scientific work. The research and development can start from early stages to prototype development. It can actually be the environment for trial and error. Unfortunately, the academy is not designed to be a business unit for manufacturing at the same time for running a business venture. But in the industry, however, can further move your prototype to a scale-up platform. And it's a different set of research and development. It's now product research and development, and it has the capacity to manufacture, to market, and to distribute. So these partnerships completes the technology life cycle from fundamental science for work to technology adaptation and commercialization. But the problem here in the Philippines is, despite of the push from one portion of the government to say, go, 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 do this, do that, but the other side of the environment is there are no adapters. They would prefer technologies coming from outside, which creates a very confusing message to the scientific community. But where is this idea actually heading as far as our advocacies here in the Philippines in pushing for local technology adaptation and commercialization? Where we believe that the first step towards getting somewhere is actually to decide that you are not going to stay where you are. So when we created the startup company, we felt that yeah, we put together an outfit to distribute, but what about those who are following us? So to continuously nurture the growing biotechnology ecosystem in the country, the first spin-off company of the university will create the first biotechnology park in the Philippines, which we call the Lighthouse Project at Clark, Subic, Pampanga. The Lighthouse Project will be the safe space where scientists, entrepreneurs, and innovative thinkers in making their scientific ideas grow into an innovative startup incorporating both the scientific and business sides of it. It will be the central hub where our researchers can scale up their prototype with all the access to the expertise and support that they need. Example, if you have if you have a light bulb idea and you need a place to make it grow into an innovative startup, but you don't know how to run the business side of it, you just come to the Lighthouse, which translates innovative ideas tomorrow for today. So if you have another light bulb idea, but you need a place to scale up your prototype, and you need access to both manufacturing and expertise in the field, you go to the same location. How do we make this all happen? The Manila Health Tech, which was created in 2013 through the University of the Philippines Tech Transfer Program, has now generated 4 subsidiary companies that does manufacturing, multi-omics diagnostic validation testing laboratories, data analytics and artificial intelligence, and alternative therapeutics for vaccines, biologics, and therapeutics to create the value chain needed by young scientists and developers to not go through the challenges we went through when we were all starting. So we built this infrastructure to allow young scientists to actually scale up their technology. So if we felt that the challenges, the heartaches of all that we did along the way can actually lessen down its burden if we help others bring their technology out to the market. We feel that it's a good start to grow this technology. So the Gen Amplify Technologies, which is now the subsidiary of the Manila Health Tech, is an ISO certified and FDA registered manufacturing facility for scaling up of all of your diagnostic platforms for both isothermal and polymerase chain reaction in the Philippines. These are the barriers we have experienced so far. And we're hoping that it's going to change as we further improve our part in terms of developing quality diagnostic technologies to be developed, to be released out there. There is perceived unreliability and skepticism. There is lack of time to learn the new technology, so which means that let's just do the old one, even if it doesn't work very well because it's we're used to it. No, lack of support from the organization or technology adapters. Our regulatory standards are requirements are very confusing. So we experienced that firsthand. One rule now, and then several months later, there's a new rule. And sometimes this policy developer doesn't understand that every changes that they do cost a lot of a lot of resources to remodify based on their new rules, If you are an organization with very limited resources, it can actually be debilitating. So it's very confusing. One side, you are being pushed to translate your technology, but the other side, you are having difficulty getting it adopted locally. So there are several strategies out there for local technology adaptation, but it needed to be implemented and enforced to incentivize and encourage tech developers to go to the next level. And these are just one of those things that we've encountered. And I'm sure from your own experience in some tech developers in the audience may have also some ideas on how to further improve local technology adaptation. My personal take on local development initiative is that it will take an entire village to create an ecosystem that will support biotechnology and economic growth in the country. Programs like the ones created by Professor Alusila is one of those things that is very powerful by creating a village of an ecosystem here and abroad. The government push for science and technology needs to be complemented with local adaptation of technology to encourage investments in the country's science and technology. If there is government adaptation of small technologies and startup in the Philippines, it will set an example to young developers to say, I think we should go into that route because the government values our scientific work. So getting grants to push for technology development is one thing and it's wonderful, but getting the government to adopt the output of this science and technology investment by their own by the government itself is another. The regulatory cycle needs to be adaptive to the rapid evolution of technology development and translation. Of course, we need to invest in our science. Thank you very much for the invitation and have a wonderful day to everyone.
Thank you very much, Dr. Destura. I really enjoyed that talk. That I've met you since 2014, and I know what happened, and through the news and through social media, and it's the first time that I saw some of the intricate things about starting Maninal Tech, or the first biotech company spin-off in UP Manila. Now, one thing I found funny, sir, would be the government is confusing. So I actually was laughing at the background. But for questions, okay, so please try to type them for our participants through the WUVA app, as well as in our Zoom link. So probably the first question by Dr. Kwang, okay. Professor Destura, thank you for your valuable presentation. I am curious about in the Light Hub project, also help the young scientists to get the investment like the angel fund. Probably Dr. Kwang is asking if for the Light Hub project, maybe you can give us more details about this, Dr. Destura.
Yes, so the Light Hub project is going to be our advocacy in creating an environment for the second generations that will follow, that will come after us. I do not want them, or I do not wish them to experience the challenges we've had that could have been reduced if such facilities or initiatives are present. So the Light Hub project is a non-profit, non-stock initiative that will be placed in a 6-hectare facility at the science and technology location at the Clark City, at the new Clark City. Its main purpose really is to create technology business incubations for young scientists who wants to commercialize their product, and can also be a platform for angel investors, for government infusions, or for private investment groups to establish the technology. It will partner with the business groups of other schools to actually utilize that as an opportunity for their business students to collaborate with the sciences team in creating business development portfolios for each of these technologies, and it becomes now a shared relationship for both. So this is a 3.2 billion peso project that's currently we are working together with local and international investment groups. So it's a long haul flight, and uh, uh, we have started the initiative last quarter of last year, and nonstop meetings and opportunities to sell the idea to a lot of investment groups who are actually very excited about it. It's too premature to talk the details about it, but basically, it is something that going to be helpful, no, in so far as the the technology adaptation is concerned.
Okay, sir, Dr. Alucilia actually is typing here some of her comments. Actually, I invited her to open her mic, but I'll just try to echo it. So, the infrastructure that okay, Dr. Alucilia is here. Yes, maybe, ma'am, you can give your questions to Dr. Destura.
Yes, so Dr. Distor Raul, I call you Raul because he's my kind of like son, right? He calls me Ate. So anyway, Raul, thank you so much for that wonderful presentation. Yeah, I truly agree with you on the challenges. Even here in the US, there are challenges, but probably there are more support compared to the United States. I mean, compared to the Philippines. So at MSU established this Michigan Biotechnology Institute to do scale-up experiments, so that if you are from the lab, they will do the scale-up. They have the infrastructure for scaling up at cost, like you pay them to do your scale-up experiments. Say if you are from a test tube and you want a 5-liter output, they will do the scale-up and work with you so that then your product will be the same as the lab product, things like that. So that's probably an area of, is that part of the plan for the Light Hub?
Yeah, so maybe I was conscious of my time, so I kind of skip that particular, but maybe this might give you an idea what the plan is, if I may share.
Go ahead, sir.
So it's basically a facility which has toll manufacturing capability. It has a logistic unit so that you don't have to spend money on storage, cold chain, and all of these things. And it has a CGLP laboratory facilities for molecular diagnostics and amplification platforms and biologics, so that it's a thematic facility. It's not specific for a certain disease, but it's a platform-based facility. So for example, if your technology is in the area of nucleic acid amplification, it doesn't matter if it is isothermally amplified or by polymerase chain reaction or a combination, that facility will be designed for nucleic acid manipulation. If you are in antigen, antibody, or immunological platform designs, that facility will be designed to actually handle this type of environment of research platform. So it's a platform-based thematic areas of laboratories, so that every technology that it passes through this scale-up units is already CGMP and CGLP grade. So they don't need to spend their resources creating this kind of facility. So when they have an investor, they would say, oh, it is just for raw materials. And then it will become a product in a few weeks because there is now a facility we can actually manufacture this without disclosing our IPs. So these are the ideas we have worked so far and consulted the science and technology park organizations in the European countries to tell us their best practices. And they've been there, done that moment to shorten our learning curve. So it's a very ambitious project. And you just have to start the first step so that you can actually get to the other side. I don't know how long it's gonna take, but I have an idea what it's gonna take and how it's gonna take. So it's easier to convince people from the outside environment because they know what it's all about. But maybe in the future, if we keep on educating our community, there may be some adaptation. So I have accepted those challenges, and I know it's gonna be very, very challenging. But for as long as there is one Filipino that will believe in what we're doing, then it's all worth it.
Well,
All
we
right.
believe in
Dr.
you,
Amelie?
so
Yeah.
we are already with you. Thank you.
Dr.
Thank you. Very good.
Destura, Vanji, because I can relate with Vanji's comment, no? And I'm quite excited, Dr. Destura. After my visit in Michigan State University last December, October, main thing that I really feel the need for the Philippines to have is the scale up. Because the information coming from Michigan State University of product of researches of the university, when it gets into the scale up, only 10% gets to the industry. And we don't have that, right, in the Philippines. And with the impressive plan that you have shown, I hope that not only one Filipino will believe in that. There would be many. It's just you need to target the right people to be in that. And one thing that I've noticed, yung scale up company that you have shown, is that part of the Light Hub already?
Now the Light Hub is a
Because there's a
different-
company already, right? That you have shown doing scale up. Would that be part of that or linked to the Light Hub?
So the Light Hub project is the CSR component of the Manila Health Tech Incorporated to provide an environment for the young scientists to thrive. Just because we went through HELL and back, to go through all of these processes, that doesn't mean we want everyone to go through the same thing. If we can make it easier for the next generation, then I think our job will become more meaningful. So this is the Light Hub Projects, is the CSR of the Manila Health Tech Incorporated group of companies, which is going to be managed by a foundation, a non-stock nonprofit. But in terms of technology support, if they need expertise in artificial intelligence and translation, the Neural Mechanics Inc., which is a subsidiary of the Manila Health Tech, has team of AI experts doing work now for both healthcare and the business sector. If there is a need for their diagnostic tests to be field validated, we have the Precision Medicine Diagnostic Facility, the Primedex Inc., that can do testings of all of their laboratory technologies being developed in the field. And if they need it to learn how to scale it up, they need help to distribute it all over. Gen Amplify Technologies Inc., which is a sales and sales distribution and marketing company, can help actually those individuals distribute and market their product. So it is not a requirement, but if help is needed, these are the outfits we created under the Manila Health Tech Group. But MTech is principally an R&D company. That's why we have diversified into subsidiaries so that they can also focus 100% in any of those areas, so that our scientists will stay and remain as scientists, not businessmen. So, and that's the purpose why the core function of the Manila Health Tech should stay as an R&D company.
Okay, Dr. Sir, I'm already extending my congratulations to you.
Thank you.
And I'm hoping that that can be echoed to other technologies, importantly on the agricultural sector, because that can be easily echoed to that. So congratulations, and I'll be supporting that, whatever support that we can give to you. Thank you, Dr. Destura. Happy to hear that. I'm really happy to hear that.
Raul, we really support that. That's a very impressive plan, very ambitious plan, but we know you have the stamina and the experience to do it. So we look forward to your success, and we are excited for you. Congratulations.
Thank you very much, Ate Vanji.
Let us know how we can help you over there.
Yeah, exactly.
Okay. Okay, very impressive and also motivating, Dr. Destura. Okay, we're hoping that this kind of project would start sana po next year, or probably in the next in the upcoming years po. Okay, now to award the certificate of Dr. Destura, so next slide please. So of course, the Global Alliance for Rapid Diagnostics of Michigan State University, of course, awards and presents this certificate of appreciation to Dr. Raul Destura of the National Institutes of Health, University of the Philippines, Manila, for his presentation entitled "The Molecular Diagnosis for Infectious Disease Program of the Philippines, Challenges and Lessons Learned over the last 15 years and Future Opportunities for Growth," at the 2023 Global Alliance for Rapid Diagnostics, or GARD Forum, with the theme "The Role of Rapid and Innovative Diagnostics in the One Health Perspective," held last March 25, 2023. Signed by Dr. Evangeline Alucilia and yours truly, Dr. Tadeus Carvajal. So everyone, let's give a virtual warm of applause to Dr. Raul Destura. Thank you very much, sir, for your time. I know you're busy po talaga, and we really appreciate you coming over and giving this presentation po.
Thank you very much, and my pleasure. Thank you.
Okay. Thank you po. All right, so we will now move on, and I know that some of you would have a lot of questions to Dr. Destura, and probably you can contact him. Okay, okay. His details are actually posted in our WUVA platform, but you could actually try to Google him as well in this case, or Google his name in this case. So we now move to our next presenter. Okay, so our next presenter would be a associate professor at the Department of Parasitology. He's a registered medical technologist, having received his BS in medical technology degree at the Manila Central University. He finished his master in public health at the College of Public Health, University of the Philippines, Manila, and afterwards he obtained his PhD in animal and food hygiene at the National Research Center for Protozoan Diseases at Obihiro University of Agriculture and Veterinary Medicine. He also received training from the Schistosomiasis Laboratory Biomedical Research Institute in the US and from the Wellcome Trust Sanger Institute in UK. He has also been a recipient of postdoctoral fellowship from the Association of Preventive Medicine of Japan and from the Japanese Society for the Promotion of Science, or JSPS. Our speaker has been conducting several researches in molecular biology, genetic studies, and the development of serological diagnostics of Schistosoma japonicum infection, both for humans and animal reservoir hosts. He has received research grants from the Global Health Innovative Technology Fund of Japan, the World Health Organization, Merck Incorporated, and DOST-PCHRD. He has also authored and co-authored several research papers on parasitology, published in international peer-reviewed journals, and is currently a review editor of Frontiers in Tropical Diseases. He is also currently the section chair of the for public health at the National Research Council of the Philippines Medical Division. He is a member of several professional organizations, including the Japanese Society of Parasitology, the American Society of Tropical Medicine and Hygiene, and the Global Schistosomiasis Alliance Research Working Group. Okay, and of course, without further ado, our my senpai, my senpai, uh, of course, Dr. Jose Maria M. Angeles. Okay, with the presentation titled "Development of Serodiagnostics for One Health Surveillance of Zoonotic Schistosomiasis." Great to see you, sir. Okay, thank you very much, po.
Thank you for that introduction, Doc Tods. So good morning, everyone. Can you see my slide?
Yes,
Yes.
po, sir, we can.
Okay. So I will be presenting our study on the development of serodiagnostics for One Health surveillance of zoonotic schistosomiasis. So just a brief introduction, schistosoma or blood flukes could cause schistosomiasis in humans, and humans will get the infection through skin penetration of the infected cercaria coming from the intermediate host snail. So the schistosome parasite will now develop inside the human host until they become adults and start laying eggs. Some of the eggs will be released in the environment, releasing together with the stool, releasing the miracidia in the environment, which will in turn infect the intermediate host snail. So schistosomiasis is also an ancient parasitic disease, as there was evidence from Egyptian mummies demonstrating the presence of schistosomiasis eggs. It is also one of the neglected tropical diseases and is the second most important parasitic disease in humans next to malaria. There are 5 major species infecting humans. However, here in the Philippines, China, and some parts of Indonesia, only the species Schistosoma japonicum is present. So schistosomiasis is prevalent in tropical and subtropical areas, especially in poor communities, and estimates show that at least 251.4 million people required preventive treatment for schistosomiasis in 2021, out of which more than 75.3 million people were reported to have been treated. 78 countries have reported schistosomiasis transmission, including the Philippines. In the Philippines, schistosomiasis is endemic in 1,609 barangays in 189 municipalities in 28 provinces in the 12 regions. And out of the 1,609 endemic barangays, 30% are classified as having high endemicity with prevalence of more than 5%, 26% with moderate endemicity with more than 1% or to less than 5% prevalence, 33% with low endemicity with prevalence of less than 1%, and 11% is still unstratified. So this data is from the Department of Health, showing results of the focal surveys done in schistosomiasis endemic barangays in the Philippines. So as you see here, there are a lot of barangays having low prevalence and even barangays with no more human cases, such as in the provinces of Bohol and Davao del Sur. So these are areas considered as candidates for schistosomiasis elimination. So for the diagnosis of schistosomiasis, microscopic detection of the eggs in the stool through Cato-Cuts technique is the easiest and cheapest method, and it is still currently being used as the sole diagnostic tool in the endemic areas in the Philippines. Although Cato-Cuts technique will give a quantitative reading on the number of eggs per gram, this technique has been reported in many papers as underestimating the prevalence of schistosomiasis in endemic areas. In fact, one study done in China, the underestimation could be as high as 56.3% for reading only one Cato-Cut slide. It was proven in another study that 7 repeated Cato-Cuts should be done to make Cato-Cuts a reliable gold standard for the diagnosis of schistosomiasis. Also, in recent epidemiological studies in the Philippines, Cato-Cuts has shown low sensitivity in examining individuals with low intensity of infection, and in using this test in endemic areas nearing elimination. Therefore, in the advent of schistosomiasis elimination, wherein most of the infected individuals will have low intensity of infection, microscopy could be used in detecting the microscopy could not be used in detecting the real infected individuals, which may cause untreated individuals and continued transmission of the disease in endemic areas. Serological tests could then be used as a replacement for Cato-Cuts in areas having low intensity of infection. So during schistosome infection, only 20 to 55% of the eggs laid by the schistosome adults in copula will be excreted in the feces. So low intensity of infection might lead to the sporadic release of the eggs in the feces. Therefore, these infected individuals will remain undetected when the feces examined does not have the schistosome eggs. Thus, the cases are called egg-negative, worm-positive schistosomiasis. In such cases, when parasitological tests could not detect eggs in the stool, serological tests seem to be more valuable in the diagnosis of schistosomiasis. Serology usually provides limited performance in acute schistosomiasis and is not capable of reliably measuring the intensity of infection or discriminating between active, past, and successfully treated infection. However, development in recent years have provided serological assays and antigenic targets to potentially fill these diagnostic gaps and improve the performance of serology. So therefore, serological tests seem to be more valuable in the diagnosis of schistosomiasis. So current ELISA for schistosomiasis either utilize crude or recombinant antigens. So crude antigens are proteins derived from the whole parasite, may it be from the egg, as in the soluble egg antigen or SEA, or from the adult parasite, as in SWAP or the soluble worm antigen precipitate. But since crude antigen contain a lot of antigens, just like it could cause cross-reaction with other parasitic infections, making it to have low specificity. So when compared with other types of ELISA, SJ-SEA has shown 71.6% and 70.7% specificity in 2 of our recent publications. So another disadvantage of using crude antigen for serological testing is that production of crude antigen is quite tedious and it takes a long time. So this is because the whole life cycle of the schistosome parasite should be maintained inside the laboratory. So continuous infection of the intermediate host on Comelania snail and the definitive host rabbits or mice is done to have a steady supply of the crude antigens. So these disadvantages in using crude antigen-based ELISA have led to the application of recombinant antigens in serological tests for schistosomiasis. Recombinant antigens are specific proteins of the parasite produced in the laboratory through recombinant DNA technology. And with the availability of the whole genome of Schistosoma japonicum, we could now do antigen mining using bioinformatic tools and genomic databases. So there are already a number of recombinant proteins being evaluated for their diagnostic potential in schistosomiasis, and because they are very specific to schistosomes, they are showing high sensitivity and specificity. So our research group has screened several recombinant antigens for their diagnostic potential in schistosomiasis, including thyrodoxine peroxidase 1 or SJTPX1 and tandem repeat proteins. So thyrodoxine peroxidase 1 or SJTPX1 is a major detoxifying agent against hydrogen peroxide in helminths and is present on the tegument of Schistosoma japonicum, making it exposed to the host immune system. And among the three TPX types, only TPX1 was seen in the excretory-secretory products of the adult worm. On the other hand, tandem repeat proteins are often targets of humoral responses both for protozoan and helminthic parasites. And previous studies showed that computational screening of genomes could identify novel genes encoding tandem repeat proteins or TRPs with serological significance from various parasites. So DNA sequence for data from Schistosoma japonicum were obtained from the genomic database GeneDB, and then we have used the program Tandem Repeats Finder to locate and display tandem repeats in the DNA sequences to identify the tandem repeat genes. And the biochemical property of each of the top Schistosoma japonicum tandem repeats were analyzed for proteins, molecular mass, isoelectric point, presence of signal sequence, transmembrane domains or GPI anchor signal, known antigenicity or function, database using both the DNA and induced amino acid sequences, and evidence of protein expression by comparison with the previous study. And also with the transcriptomic expression levels by comparison with the NCBI EST database. So on the basis of this analysis, 4 tandem repeat genes were selected for production of recombinant proteins. So there are 4 tandem repeats that were selected for serological evaluation, namely SJ1TR, SJ2TR, SJ4TR, and SJ7TR. So a statistical analysis of the ELISA results for human has shown that SJTPX1 and SJ7TR have high sensitivity and specificity. So if we look at the results of the ELISA for these 2 antigens, when tested with samples from treated individuals, they are showing negative results. So this proves that SJTPX1 and SJ7TR could only detect active infection and not the past infection. So also cross-reaction with other parasitic infections such as opisthorchiasis and paragoniomyces were only seen in SEA or soluble egg antigen, but very minimal in SJTPX1. Also, Schistosoma japonicum is zoonotic in nature. So as it is known that Schistosoma japonicum could infect not just humans, but also at least 46 other mammals, including the cattle, water buffaloes, rats, dogs, and pigs. So this has led to the current control problems for zoonotic schistosomiasis. So the current fragmented framework of health governance for humans, animals, and environment, together with the conventional linear approach to solving current health problems such as schistosomiasis, is failing to meet today's health challenges and is proving unsustainable. So a paradigm shift is urgently required to sectoralize human animals to take a more integrated One Health approach for schistosomiasis. So for schistosomiasis, One Health approach is necessary to involve the importance of the animal reservoir host in the disease control. So if we look at the current control strategies implemented for schistosomiasis, including health promotion, mass drug administration or MDA, diagnosis and surveillance, water sanitation, and hygiene or WASH. These are all directed to the human host. So if we improve the current schistosomiasis control strategy, we will have efforts directed to the animal reservoir hosts, such as diagnosis and surveillance and veterinary interventions, and also to the snail intermediate host via focal snail control. So our group has been working with zoonotic schistosomiasis for many years now, and we have stressed in our previous publication that the need to include animal surveillance in the elimination guidelines for zoonotic schistosomiasis, particularly using water buffaloes as the sentinel animals for elimination surveillance. So this will prevent emergence and reemergence of schistosomiasis in areas free of this parasitic disease. So why did we recommend water buffaloes? So if we compare them with other animal reserve hosts for schistosomiasis, we could see that although it exhibits self-cure phenomenon, its continuous exposure in the transmission sites due to their agricultural functions made them steady sources of the schistosome parasites. In addition, in China, environmental contamination of schistosome eggs is highly attributed to bovine defecation due to due to the massive size of their excreta. This is also supported by molecular studies, and these reasons made water buffaloes very appropriate sentinel animals for surveillance. So we have also tested our recombinant antigens for these water buffaloes, and statistical analysis of the ELISA results for water buffaloes has shown that SJTPX1 and the tandem repeat SJ1TR have high sensitivity and specificity. So results in the serological assessment for both humans and water buffalo show that the best recombinant antigen for both is SJTPX1. And while for the tandem repeats, SJ1TR is good for water buffaloes, while SJ7TR for humans. So these differences in antigenicity can be explained by the differences in the immune responses among various host species. So as a follow-up to those results, we have conducted another study that aims to test the field performance of the recombinant antigen ELISA in 4 endemic municipalities in the Philippines. So we have collected serum and stool samples from water buffaloes in the highly endemic Catarman, Northern Samar, in the moderately endemic Calatrava, Negros Occidental, and in two municipalities that have no longer cases of human schistosomiasis since 2012, which is Talibon and Trinidad. So we did RA ELISA and SEA ELISA, while for the stool we did microscopy and PCR. So here is the tabulated results of the positivity rates of schistosome infection in the 4 endemic municipalities. So as expected, high positivity rates were observed in the highly endemic Catarman and moderately endemic Calatrava. Also marked positivity rates were seen in Talibon and Trinidad, where no human cases of schistosomiasis were reported. So our results indicate a marked positivity among the water buffaloes tested in near elimination areas in Talibon and Trinidad, where no human cases have been recorded since 2012. The presence of this, the schistosome parasites in the bovine host poses a threat in the human schistosomiasis elimination, and this threat can possibly be avoided by setting up a sensitive animal surveillance system and can detect infection in animals for prompt intervention. Hence, animals serving as sources of infection can be treated properly, preventing any further contamination of the environment with the schistosome eggs that can start transmission anew. So the intensive mass drug administration done in the endemic areas have resulted to infected individuals to manifest low intensity of infection. So this greatly affects the diagnostic performance of CATOCATs and indicate the need for a more sensitive diagnostic tests to be used in such conditions. So our results showed that microscopy has the lowest positivity rates among the water buffaloes in the study areas, and RA ELISA should be included in the animal surveillance of the National Schistosomiasis Control Program to ensure elimination and prevent emergence and reemergence of this parasitic disease. So in collaboration with the US-based biotech company InBios, we have tried to apply one of our recombinant proteins, SJTPX1, in immunochromatographic test format. So although the results seem promising, this still needs more improvement and optimization. So we have tried two prototypes of the POCT. One is IgG detection format and the other is IgG4 detection format. And the prototype in IgG detection format has so far achieved the highest sensitivity among the two, which is 73.1%. So unfortunately, this project has been stopped due to lack of funding, but our group will revisit the transformation of the diagnostic tool from RA ELISA to paper-based rapid diagnostic tests in the near future. So, and finally, when we talk about surveillance system for One Health, it should employ a unified medical and veterinary diagnosis using reliable and accurate diagnostic tests. So this should be done in collaboration in between the human health sector and the animal health experts in the implementation of the surveillance system in schistosomiasis endemic areas in the Philippines. So I would like to thank our funders, GHIT Fund, Japan Society for the Promotion of Science, WHO Western Pacific Region, and the Tropical Disease Research. Also to my research group at the Department of Parasitology, College of Public Health, University of the Philippines, Manila, our collaborators from Japan, University National Research Center for Protozoan Diseases, Obihiro University of Agriculture and Veterinary Medicine, the University of Tokyo, and Tokyo Medical University, InBios, and the Department of Health Philippines, and all of the endemic municipalities in the Philippines where we have collected samples and tested our serodiagnostic test. So thank you for the invitation and thank you all for listening.
Okay, thank you, Dr. Angeles, for that wonderful talk on serodiagnosis for schistosomiasis. Dr. Carvajal asked me to take over for the Q&A portion for this lecture because
Okay.
he has.
I'm sorry.
Okay po.
Okay
Okay
na.
na po.
Okay, yes.
Okay
Okay,
po.
I'm sorry. All right. So thank you, Dr. Joma, okay, for the wonderful presentation. So we're opening the questions and answer portion of our speaker for today. So, okay. So, Doc Joma, for schistosomiasis in this particular case, so in your presentation, you are developing po mga yung mga zero diagnostics, right? So what are the challenges right now that you are facing in terms of developing those zero diagnostics tests?
Yeah, one of which is of course the securing samples, the positive samples from the endemic areas. That's one of the challenge. But right now we have a lot of good characterized samples in our laboratory, but we need to store it and in a very good way so that we could use it in developing different tests. And then also the acceptance of the people in the endemic areas of the test. So because this based on blood, this is based on blood, so we need to collect blood from the patients or individuals living in the endemic areas.
All right, thank you, Doc Joma. I know positive samples po talagang mahirap, and I would know that for sure. And securing that for for proof of concept, validation, and confirmation, taga po getting samples, and I think Pa would also know that in this case. So we have first Dr. Divine Ameline. Sige po, ma'am.
Thank you, Dr. Angeles, for that very nice presentation. And I don't have a question. I just have to let you know that we have the NICER program on Center for Vector Control and Schistosomiasis will be included there. We are running second year now, so would like to invite your group to the center, and let's have a talk later, no?
Yes.
Very soon, let's talk so we can already look into how we can help on the vector control, on the snail control. Okay, thank you. We'll contact you very soon, Dr. Angeles.
Sample.
So, and then Dr. Alucilia, sige po.
Dr. Angeles, thank you so much for a great presentation. So I have a question, because that you said it's very hard to get blood because now you have to go through IRB, right? You have to make sure that nobody gets infected or something like that. Have you thought about non-invasive samples? Because they would be easier to collect, firstly, and secondly, the IRB would be less stressful for that.
For schistosomiasis, there's a test for urine, but it's a POCT for urine, but it is not. It was tested using some samples for Schistosoma japonicum, but it's not showing good results. But of course, it will be best if we could use urine as a sample for the ELISA. But right now, blood serum is the best samples we have, that we are getting the best results for our test.
So could it be because there's more of the that's a parasite, right?
Yeah.
There's
in
more parasite in the blood?
schistosomiasis, yeah, in schistosomiasis, usually the adults are not that many, but there are many eggs. So a very good antigen is coming from not just from the adults and from the eggs, because the eggs are the ones that are disseminated in many parts of the body, including the liver and the intestine. So they are producing more antigens to the that are eliciting antibody response. So
But you have also the opportunity, so for the urine, I'm just looking at the urine, that to have higher volume, and can you bring that volume down? So basically you concentrate
yeah.
your target, because now you have the opportunity to get a higher volume sample.
Yeah, I think we could check that in the future, Dr. Alucilia. Thank you for that very nice thought. We could also check the other non-invasive samples for schistosomiasis.
Thank you, because it's always easier to get your samples, to get patients to submit their samples if you don't poke them. You start poking them, like, oh, that's a different story, right?
Yeah, thank you, Dr. Alucilia.
So I also in the chat box, Dr. Angeles. So Pao, okay, I think one of your students, okay. So tackling disease elimination is hard, especially for schistosomiasis, since it has several reservoir hosts. For diseases like this, sir, how would you assess or identify which is the most important reservoir host to target for interventions?
Yeah, of course. For schistosomiasis, it's not that hard because water buffaloes are in the transmission site. So that's one criteria. It should be exposed to the pathogen or to the parasites. Number two, it should have a high infection rate that is contributing a lot to the transmission of the disease. And number three, of course, it's the access to these animals if you want to test them. Because for schistosomiasis rats, it's not that accessible for us to check. It's not easy to catch rats.
Yes,
So
that's correct. Yes, mahirap po talaga. Yes. Okay, I think that for schistosomiasis, sir, because you're concentrating more on hosts that are actually being infected. Therefore, blood would be necessary for your ELISA test. But sir, in your current research endeavors, are you planning to go more for environmental samples, like maybe examining certain water samples where oncomelania would be, and try to test if whether, for example, there is high, of course, high number or high abundance of the I think metacercaria, or probably the larval stages of your of schistosomiasis po.
Dr. That is not one of our research topic or research because I think there's already one doing that in UP Diliman. So
Okay, I understand po. Okay, yes, yes.
environmental DNA.
Yeah, this is actually from Dr. Padilla, I think, if I'm not mistaken, or basta po, yes, I remember po that. But you're working with him po on this part, because these kinds of tests would actually try to allow you how the transmission dynamics would occur. You have the environmental DNA, and then looking at on the prevalence or infection rate of your maybe hosts, like such as your buffaloes on this way. Now, sir, you just going back to Dr. Alucilia, because urine you said is one possibility, but does it what are you trying to detect on the urine? Are you detecting eggs on this case? Normal in certain
No.
situation. So what are you detecting in urine that would let the parasite pass through the urinary tract? Because I know it's going to be more of blood or intestinal po, di ba? In this case. So there po.
So in the test available now for schistosomiasis, which is testing the urine, they are checking the circulating antigens. So these are antigens from the parasite that circulates together with the blood and then filtered to the kidney and then pass through the urine. So they have found that that is very detectable in urine, the circulating antigens. But for us, we are checking the antibodies, so we are not so sure yet if the antibodies are also they are also present in the urine.
Okay. Okay. Yes, Dr. Alucilia.
Yes, so if that is the case, there's a possibility that there could be schistosomiasis DNA floating in the urine. Could that be possible?
Yeah, it's possible, but the detectable amount is the one that is very critical. How to detect such little DNA.
And that is where you can concentrate. You can develop a concentration method. And the reason we're saying that because we have a magnetic nanoparticles, and TADS is aware of this one, where you can use it to basically attach the DNA fragments, adsorb the DNA fragments to that magnetic nanoparticle, because they are electrically active. And so then once you pull out, you will have the DNA sticking to the electrically active magnetic nanoparticles. And then you can extract it, get a high volume of urine, put the magnetic nanoparticle, use a simple magnet, and then you can concentrate your DNA fragments. That may be an option.
Yeah, that is a very good idea, Dr. Alucilia.
TADS and Divine know about this magnetic nanoparticles.
Yeah, we could work on that.
Yes.
Yes, Banji, I was thinking about that on the vector level when I'm talking to Dr. Angeles on that. So that's why I would like to invite the group. No, we can actually, this is a nano diagnostic using the technology developed by Dr. Alucilia, but now it's on the higher level organism. We're looking into the microorganism, but I think we are also talking. We had an initial talk with Banji on how to use this on a higher level organism, just like the parasite. So I'm quite excited on this, because on the vector part, no, and because we need to control first on the vector to limit effect on the human population and also the livestock on this. So that's why that's what I was thinking about when I invite Dr. Angeles to join us. The higher level organism detection using the nanodiagnostic technology developed by Dr. Alucina. So
Yeah,
lalo
so,
na ako na-excite. More excitement for me.
so Divine, you can use the nano biosensor for the higher level, but you
Yeah.
can also use the electrically active magnetic
Mhm.
nanoparticles to capture his DNA, a floating DNA in the urine,
Yeah, yeah.
because that electrically active is positively charged. Yeah, positively charged.
Yeah,
And
yeah,
then the
yeah.
DNA is negatively charged. It will attach. It will like
Yes.
absorb that to the
Yeah.
nanoparticles.
Okay, I think we have we will have something to cook for this particular for this research topic, no? To help in the control of schistosomiasis, looking into the 56% already, no, na cases. Sabi ko, I felt so. I think there's a need to work on this. It's not anymore a neglected tropical diseases, it becoming an important disease already in the Philippines. So thank you. So let's Vanji, our group, you know, with Dr. Angeles and UPLB group, Adia, we can work together on this with Dr. Angeles. Thank you.
Okay, okay. So thank you, Dr. Divine, and also Dr. Vanji. Okay. So of course, if you need, if you have more questions for Dr. Angeles, you may contact him through email. Okay. He's and of course you try to Google his name as well for his contact email address. Now, thank you very much, Dr. Angeles. So we now move forward to the awarding of the certificate. So the Global Alliance for Rapid Diagnostics of Michigan State University awards this certificate of appreciation to Dr. Jose Maria Angeles of the College of Public Health, University of the Philippines, Manila, for his presentation entitled Development of Zero Diagnostics for One Health Surveillance of Zoonotic Schistosomiasis at the 2023 Global Alliance for Rapid Diagnostics Forum, the role of rapid and innovative diagnostics in the One Health perspective, held last March 25, 2023, signed, of course, by Dr. Evangeline Alucilia and yours truly, Dr. Tadeus M. Carvajal. So let's give a virtual warm of applause to Dr. Jose Maria Angeles for sharing his presentation to us today and giving us opportunities to do collaboration for an important neglected tropical disease. Thank you, sir, for your time. I really appreciate it po. Okay, we're hoping for future collaborations with you and your laboratory. All right. So we now move to our last speaker. And of course, we had a lot of good presentations okay, in the past 1 or 2 hours on this. And we learned a lot in this case. So this next speaker of ours, of course, I already know him because of our affiliation with the Philippine Society for Microbiology. And actually, his topic is actually very interesting in this particular case. So he is actually a professor at the Graduate Institute of Animal Vaccine Technology and the International Program in Animal Vaccine Technology of the National Pingtung University. He's also the director of the Companion Animal Research Center of the National Pingtung University. He took his bachelor's degree in microbiology at the Soochow University in Taipei, Taiwan, and received his master's degree on microbiology and immunology and PhD as well on basic medical sciences from the National Cheng Kung University in Tainan, Taiwan. His research lab focuses on developing fast detection kits against viruses, vector-based, exosome-based, exome-based vaccine development, cytokine and transcriptome analysis, and the development of antiviral treatments. Our speaker has several patents to his name and has authored and co-authored numerous publications relating to the field of immunology, biotechnology, and molecular diagnosis. Without further ado, I would like to invite Dr. Kuo-Ping Chuang, a professor of the National Pingtung University of Science and Technology, and with his presentation titled The Rapid Diagnostic Kits Development for Companion Parrot and Pigeon Virus. Thank you very much for accepting our invitation, Dr. Chuang, and you may start with your presentation.
Okay, I think first of all, I want to thank the invitation from the GARD and thanks for the introduction of Kavaja professor. And on the other hand, I also want to thanks for Professor Ronino because we will initiate a new project about to develop the fast diagnosis of some diseases. Okay, so now I want to share my screen. Okay, can everyone see my PowerPoint? Hello? Hello?
We cannot see the PowerPoint, sir.
Cannot? Oh, gee, this one.
Cannot see it, Dr. Kwang. Yeah, there you go.
How about that?
It's okay now.
It's
Okay. Okay. I'm so appreciated to talk this topic in this conference. Okay, so the title is the development of rapid diagnosis kit for companion birds. I think in the beginning, I just want to talk about the topic about rapid diagnostic kit for companion animal. But on the other hand, in the end of this talk, I also want to introduce this companion animal research and the other research we have done in our lab, okay? So in the beginning, we want to talk about these two viruses. I think maybe some people are familiar with this virus we called pigeon circle virus, the PICV, yeah. And the PICV belong to circle virus. And on the other hand, we also focus on a disease from parrot called BFDV. And we can see this BFDV virus causes the feather loss and the beaker morphology change in parrot. So I think these two viruses is very important for the pigeon and parrot. And we also know the pigeon racing or the parrot is a very important bird, no matter in Taiwan or over the world. So that's why we focus on these two virus disease and want to develop the rapid diagnosis for these two viruses. Yeah, and wow, what happened? Okay, so first of all, why we chose this pigeon? And the first question is, do you know how much production value of the Russian pigeon markets is, right? So we will see in this map. I think in the first step, we focus on Taiwan, right? Because there are more than 100 racing game per year, and the total bonus up to 100 million US dollars. Okay, this is a very big market. And on the other hand, in China, the bonus also over 70 million US dollars. And this situation in Asia is very huge and very incredible, right? But on the other hand, no matter in Europe or North America, also have the pigeon racing, and the market is also very huge. And until now, there is no fast diagnosis or treatment for pigeons disease, okay? So that's why we want to focus on this disease. Yeah, right. So this is National Geographic Journal, and he have a special book to mention about the Taiwanese pigeon racing, okay? So the Taiwanese sport of pigeon racing is also very well known through all over the world. There are approximately 500 pigeon racing club, and 50,000 pigeon flyer residents in Taiwan. And most popular or most well known is the prize money is so great. that no country in the world can match it. We just see that, right? And there are two special characteristics of Taiwanese pigeon racing. One is the oceanic racing, because we bring the bird to the ocean and then release bird and fly back to the Taiwan, okay? And on the other hand is we use the young bird, or we only use the 6 months bird for racing competition. So that's why, because we only use the young bird for racing, and the young bird and the racing is pressure. So that make the pigeon to get the disease very easily. So that's why the pigeon disease is very important in Taiwan. And later we will talk about the pigeon disease. What kind of pigeon or disease or infect the pigeon? First of all is the virus disease. They are including adenovirus, paramyxovirus, rotavirus, pigeon pox virus, circle virus, okay, and pigeon herpes virus. There are so many kinds of virus can infect pigeon. And they also have bacteria disease, including Salmonella, Escherichia coli, Mycoplasma, or some protozoa, just like a coccidiosis, Hemoproteus, or Trichomonas, right? And some mycosis disease, like the Candida albicans. So there are many kinds of disease, yeah. But just like I mentioned until now, there is no effective or marketed diagnosis kit for pigeon, right? Stuck. Okay, first, we introduce the structure of the pigeon circle virus. The pigeon circle virus belong to the Circoviridae family and the circle virus genus. This virus belong to non-envelope virus, just like here shows, and the structure is icosahedra, right? And the most special characteristic is that the genome are circular and non-segment, and the diameter range of the circle virus is from 70 to 22 nanometers, all right? And the most important is this virus will cause immunosuppression of the pigeons. And we will know if the pigeon's immune system was disrupted. Okay, there are many kind of secondary infection will cause disease. So that is the original of disease of pigeon. So is to detect is very important to this virus or for the healthy of pigeon, right? And later we will talk about the parallel, and we can see here, Taiwan here, and Taiwan here. So we can see the Taiwan is the major location to export the parrot. We can see here the blue means export quantity, and the red is the import. So we know Taiwan is very important place to export parrot to all over the world. And we know the parrot trade is very common all over the world, but the parrot is protected by CITES, the international organization, right? But now because the older people get more and more all over the world, right? And the parrot can talk, can communicate with human. So that's why there are more and more people like to raise the parrot to stay with them when we are old, right? So we can see there are many people like the large parrot, just like I mentioned, because the parrot can communicate with people, and they like children can talk with you, right? So this is why parrot trade is become more and more important, and Taiwan is a very important place to raise or to export the parrot. So that's why we need to focus on these two kind of species. And on the other hand, we also focus on circle virus. Okay, and BFDV and pigeon circle virus also belong to circle virus. Okay, and here we also can see, sorry for the figure is Taiwanese, but here we can see the yellow is the export money and the blue is the import money. We can see the import money is decreased in Taiwan every year. It means when we import the parrot into Taiwan, and then we start to breeding, right? And we breeding by ourselves, and that we don't need to import again. But when we breeding more and more, the export also increased. Yeah, about 50 million USD dollars per year. So I mean is the parallel trading is very important for Taiwan, and the money is very huge, right? And on the other hand, the pigeon also a very important animal in Taiwan. Yeah, and just like I mentioned, the parallel BFDV also belong to Cercoviridae family and Cercovirus genus. So they are almost the same, right? And this virus belong to non-enveloped icosahedra with a complete genome size of 2 kb single-strand circular DNA. And just like I mentioned, the diameter is 14 to 70 nanometer. And this is the protein structure or the structure of this virus. Because in our development strategy, the structure is very important. Because in the old way, people need to express the protein and then to produce the monoclonal antibody, and then use this monoclonal antibody for rapid test development. But in our lab, in the first step, we will analyze the protein, okay, the hydrophobic or hydrophilic. Then we will design the synthetic peptide by protein structure. And then we directly use the synthetic peptide to inject into the mice and produce the polyclonal antibody, and then use the polyclonal antibody to produce the rapid diagnosis kit. So by our method, we can reduce the time. Okay, we can development the kit in almost maybe 2 or 3 months, and we can get a very good result. So later we will talk about that. So in our project, the structure of the virus is very important, right? So the BFDV can cause acute and chronic disease of the parrot, okay? After the parrot infect with the BFDV, the virus causes acute infection, okay? Under 3 years age. During the first long feeder period, or just like 28 days to 32 days old, the birds will be depressed and shed their feathers, just like the figure shows. The feeder are losses from the parrot, and the virus also causes septic, pneumonia, and enteritis. Okay, and then this symptom will lead to death in severe case, but most of the deaths were caused by secondary infection. Just like I mentioned, because the circovirus infect the bird and destroy the immune system, then there are so many kind of there are so many kind of secondary infection will cause the death of the parrot. On the other hand, the virus also cause chronic disease, just like occurred after molting, with chronic progressive symmetric poor feeder growth and occasionally bigger lesion. Because we also can see here, this parrot's beak also, the morphology of the beak also disrupt, so that parrot cannot eat anymore. So the parrot will die, okay? So this disease is important for parrot all over the world. And then we comparison the virus detection method. I think there are many people talk about a LAMP, and I think LAMP is also a very conventional and cheap method, but LAMP still need the DNA isolation and some technique scale. So I think maybe farmer or the owner or the animal owner are not very easy to do the LAMP by themselves, okay? And just like this table shows, there are so many kind of method can be used for virus detection, including the histology, ISH, dot blood, hybridization, PCR, ELISA, or our target strip. You can see the strip is very convenient, right? You just need to waiting for 10 minutes and everyone can do this experiment, right? But the most disadvantage of strip is the sensitivity, because I think everyone knows the major for the strip is the antibody binding to the antigen, right? But the interaction between antibody and antigen is not so sensitive, but the PCR is more sensitive. So this is the most disadvantage of the strip, right? And the other advantage, including the waiting time, strip also have the advantage, and person training and the cost also very cheap. So that's why, just like the COVID-19 pandemic, everyone use the strip at home, and it's very easy, and you only need to wait for 5 to 10 minutes, okay? So that's why we chose this method for the fast diagnosis of these two diseases. But on the other hand, we also have some publication about to use the lamp for these two diseases, okay? But in this time's presentation, we will talk about the strip, okay? So the next step, I will introduce the competition method for the strip development. First of all, we can see here, okay, in the right side, okay, this is the competition method. And the advantage of the competition method for strip is we only need to produce one antibody, right? And this method's theory is we have the protein, okay? And then protein labeled with the colloid gold. And then the protein put in the sample pad here, right? And we generate another antibody, and antibody will code on the test line, okay? So if the sample contain virus, right? The virus will compete with our protein, okay? And the protein cannot be binding with the antibody, right? So there is only one line, one control line, right here. But if the sample don't contain the virus, right, the protein with colloid gold or binding to the antibody. So there are two lines. It's different. It's totally different with the sandwich method. Okay. In sandwich method, we need to develop two antibody. One antibody is coding on the test line, and one antibody will code on the sample pad, right, with the colloid gold. So if the sample contain virus, the first antibody with colloid gold or binding to the binding to the first antibody. Oh, sorry, I'm wrong. This one is the sandwich. You can see there are two antibodies. Sorry. So the first antibody will binding to the virus, and then the virus will binding to the secondary antibody. So there are two lines for sandwich. It means the sample contain virus is positive. But if the sample contain the virus will compete with our protein, right? And the virus not binding to the antibody. So there is only one line. So this is confirmed. So if you use the sandwich or computation method, the result is totally different, is opposite. So just like I mentioned, why we chose the computation method, because we only need to generate one kind of antibody, okay? But if you want to use the sandwich method, you need to generate two kinds of antibody. One is here, the other is here, right? And on the other hand, we can synthesize our peptide. It's very easy and very cheap. You can synthesize the peptide and code with the colloid gold and then generate one antibody, and you can have this test kit, okay? So that's why we chose this method, right? So the first step, we need to analyze the property of our protein, okay? The first priority is we need to choose the hydrophilic protein, okay? Because the hydrophilic protein is outside, okay? Only the protein located outside of the virus can be recognized by the antibody. So in the first step, we analyze the hydrophobic or hydrophilic of the protein from PICV or BFDV. And we can see the yellow one is the hydrophilic protein, right? So we can see in the capsid protein, there are 7 hydrophilic regions. And the next step, we need to analyze the structure to check the location of our protein is outside of the virus, okay? So we can see that this is the subunit of the capsid protein of circovirus, okay? And this figure shows 3 capsid protein binding together, just like this pink one here, okay? And we can see the yellow, red, and blue. These 3 peptide is located outside of the virus particle. So in the beginning, we chose 3 protein for polyclonal antibody production and then test which region is the best one, have high sensitivity and can have a very effective for the detection, right? So just like I mentioned, first step, we need to make sure the protein is hydrophilic. And then we need to know the protein is located outside of the virus particle and can be recognized by antibody and can be used for the strip development, right? And then we synthesize the peptide and inject to the rabbit for several times to produce the polyclonal antibody. And this is the schedule for the polyclonal antibody production, okay? We inject 100 microgram per microliter of each peptide to the rabbit for 2 times. And then we detect and sacrifice the sacrifice the rabbit to get the polyclonal antibody. After rabbit produce polyclonal antibody, we need to purify the polyclonal antibody by protein A column. So I think everyone knows the purification process. The first process is the protein A bead coating with some protein A, right? And added your serum and then wash and elute to get the purified your antibody. So now we have the synthesis peptide and polyclonal antibody against this peptide. And the next step is we need to check the polyclonal antibody can recognize our peptide, okay? So we use the dobra to get the result. First of all, we put some different concentrate of the peptide. We call the BVFD 235 to 249, the peptide on the memory, and then use the rapid produce antibody to recognize this protein. And we know, okay, our polyclonal antibody is very sensitive, even there is only 0.1 nanogram of the peptide can be recognized by our polyclonal antibody. So until now, we make sure our peptide is working. And we also know that our protein, our antibody is working. So the next step is to produce the strip and then compare with the qPCR. And we will know, we collect 12 sample from parrot and then detect by qPCR and strip. And we will know here, only 1 sample is different between qPCR and strip. Okay, so after calculate, we know the sensitivity of our strip is 100%, but the specificity is 80%. Okay, so I think this sensitivity and specificity is enough for companion animal or for parrot. So we have technique transfer this technique to a company, and later we will talk about that, a very interesting story about that, okay? And there is a video to show you the really product of our product, okay? I think everyone knows it's very simple. You just put the sample into your strip and wait for maybe 1 or 2 minutes. You will see 1 band or 2 band to make sure the parrot infected or not infected with the virus. Yeah. I think. Yeah. You can see there are 2 band and there are 1 band. And because our kit is competition strip, so this one is positive. sorry, this one is, yeah, this one is positive. Sometimes I also confused by the two band, okay? Yeah. Okay, so this is our research about this virus. And just like I mentioned, we also established a companion animal research center, okay? And in this center, we primarily focus on fast diagnosis, development, and treatment for virus infection and immunological related disease for companion animal disease. So in the future, maybe you have the chance to go to Taiwan and go to MPUSD. I think you can visit this center. Okay. I want to talk about this center because we have the scale up factory. We can see here. This is the factory for this center. And this factory is GMP scale up factory. So we have this factory to help us to produce the protein or produce the monoclonal antibody in GMP regulation. So after we have this factory, scale up factory, we can easy to scale up and then transfer this technique to a company and to establish a new company. Especially, we transfer this technique to our student, to our PhD and master student, okay? They establish a new company to sell our product, okay? And, sorry, it's stuck. Okay, so the major goal of this center is we called total solution for companion animal disease, because we know diagnosis is very important. But after diagnosis, if the animal not infected with the pathogen, what should we do? We need to develop the vaccine to immunize the animal to prevent infection in the future, right? And if we know this animal is infected with some disease, especially for virus disease, what can we do? We need the treatment for the animal, right? So that's why we call total solution. After diagnosis, we need a vaccine or we need treatment for this animal. So in this center, we also focus on vaccine development, diagnostic kit development, and treatment development, okay? And in the end, we will use the big data to analyze the gene, okay, about parallel pigeon, because we want to select the high performance pigeon and help the farmer to breed the good parrot. Yeah, right. So this is the goal of our center. So I just want to show you another research of our center. This is a kind of virus infection of parrot. We can see the stomach is swollen, okay, significantly. And in this center, we also developed some interferon. And after interferon treatment, we can see the stomach is become normal. Okay, so this is what I called total solution, because we know the parrot infected with virus, and then we treat with the another product, interferon. So, and then the parrot become healthy. We also developed some product. This is the photo of the product for the skin problem of dog. And you can see there are many red spot, and the hair is lost of the dog. And after treatment for 1 week, you can see the hair start to grow and the red spot is disappear, okay? So this is our another research. And we also have the histopathological analysis of the treatment. In this left figure, we can see there are many eosinophils. It means the skin of the dog is inflammation. But after treatment, the inflammation was inhibited and become normal, okay? So this is the mechanism, our product to inhibit the inflammation and make the red spot disappear, okay? We also test for some feline virus infection, okay? This is the pre-treated condition. We can see there are many scars because this cat infected with the feline leukemia virus. And this leukemia virus will cause a lesion and the body fluid will continue to outside, right? So there are a lot of scar in the cat. But after treatment for 5 days, you can see the hair start to grow and their scar is disappear, is very significantly. Okay. We also use the interferon to treat the virus infection of cat, right? And I think many people see this figure several months ago. We also use this interferon to treat the pigeon. And you can see before treatment, the pigeon is depressed, but after treatment, the pigeon become very happy and look good, right? And on the other hand, the other pigeon also look not good, but after treatment is very superior. Okay, we also detect the virus level of this pigeon. We can see after different concentrate of interferon treatment, the decrease fold of the virus is dramatic, all right? According to the time, the day post-infection, right? The virus will be decreased. So we know our total solution from diagnostic kit or treatment is okay for this companion animal. So until now, we established a company we call Serapha. And the student, this is my PhD student, and this is the master student. These two students open this company and then go to the competition of Taiwan. And they win the 2020 new company competition in Taiwan, right? So now they sell this product, interferon, and sells the circovirus test kit, including the kit from pigeon or the parrot. They also sell the product to inhibit the inflammation condition of the companion animal, right? So in the end, I want to thank many students and many faculty from all over the world, including the John come from Vasinova in Japan, and Professor Saito from Miyazaki University, and Professor Maria from Malaysia, and another professor come from University of Philippines, Maria, and the other person, Chia Ching, come from the Singapore. Okay, so thanks for your listening. I hope you like this topic. Yeah, thank you.
All right, thank you, Dr. Kwang. Okay, very interesting presentation actually, and very impressive also with the centers, with the center that you actually presented a while ago. Okay, I just want to ask a question about the pigeon strip test, Dr. Wang. Have you done this already on a field setting? Probably, for example, trying to use like samples from the field. So normally, most diagnostic tests, they would actually use samples coming from their laboratories, or probably from those that they have in cage, or probably that they are in a animal house. But have you tried them, like using them actually, and having ordinary people use this particular strip test?
Okay, you mean do we test this kit in field?
Yes, yes.
Okay, okay. Of course, we test the kit in field. Actually, we have sell the kit in the market now.
Okay. Oh, that's
Yeah. I think we have, how to say it, we have launched, or we have sales the pigeon circovirus fast diagnosis in the market. And this year, we also want to sell the parallel fast diagnostic kit in the market this year, right? Okay.
Is there any challenges that you had, because right now you're translating it into to commercialization? So maybe you can share to us, like Dr. Destura actually was giving out, like, his experience on trying to commercialize this particular kit in Taiwan, or probably what is the present situation there, if ever that such kit that you have would be commercialized?
Okay, I think every country face this problem very difficult, right? So that's why I ask Professor about the Light Hub. Does the Light Hub provide the funding, just like the Angel Fund? Because during my experience, I think the funding is investment or funding is very important for the company initiation, because I think we are all professors. We are all focused on research. So we have a very good research and we have a very good product, but the initiating funding for the company is not our strong part, right? So we need another person to help us to get some investment or funding to initiate. And the other thing is, I think the scale up factory is also very important, because I think in our lab is a small scale. But if you want to sell your product to the market, I think scale up is very important. So I think we cannot do everything by ourselves. Okay, my project or I am good at research, but the scale up or investment is not my job. So in our school, in our university, or in our government, we have a scale up facility, and someone can help us to find the investment to help us to have this product. Okay, this is my experience.
Okay, well, very okay. Just
Yeah.
like Dr. Destura in this particular case, also somehow similar in this. We have also Dr. Divida Amalin, ma'am. She's raising her hand.
Yeah.
Yes, Dr. Kwang, very nice presentation. I'm really, I mean, I know little about the pigeon and the parrot, but the technology, the system, it's fascinating. Your presentation is clear, and I might visit you because I'm going to Taiwan probably the end of April, and so I will communicate with you if we have time to visit your university or your lab. My question is on the Scale Up Center. It's government, you said, right?
Yes.
Is it government? And yeah.
Sorry. You first.
And the investor would be other people or students. Do they pay the Scale Up request that they will do in that center?
Okay.
Who fund the Scale Up? Because it's expensive. It's expensive to scale up.
In the initial step, the government pay money to establish the scale up factory for 1 or 2 years, and then the government ask our center to earn money and pay by ourselves. So that's why the professor need to pay for scale up. But I think there maybe have some misunderstanding, because in our scale up facility, just like my protein production, there are 5-liter fermenter, 50-liter fermenter. So you can decide which scale you want to produce. I think if in the initial step you use 5-liter, I think is enough for several batch production and is not so expensive, right? So that's why we need to buy a lot of equipment by government in the beginning. But later, the center, I need to pay to the center to scale up. And then
you pay.
Yeah, I pay.
Personal payment.
Pardon?
Personal
No. I need to find some collaboration from company. Because our school have an incubation center to help professor to find the company who are interested in about your project. So in the initial step, the company also only need to pay very few money to help you to scale up, and then we can test our final product, and then the incubation center of university will help us to find a big money from the investment. Yeah.
Okay. All right. That's clear enough.
Yeah.
But thank you very much. I'm quite interested with that.
Yeah.
And I'm hoping to visit your lab or your
I think very welcome for for you. Yeah. And I think if the other people also interested about collaboration or research, we also welcome. On the other
Yeah.
hand, I also want to announce, we also want to recruit some students for internship, master degree or PhD degree in our center to focus on this total solution method. Okay.
Okay, Dr.
Yeah.
Carvajal, there's a volunteer
Yeah,
Yes,
here in the
okay.
Philippines.
So if you have a student want to study abroad, okay, welcome. Yeah. Yeah.
Okay, all right.
Yeah.
Thank you,
Okay.
Dr. Kwang.
Yeah.
I'll communicate with you, okay?
Okay, no problem.
Okay.
Thank you, Dr. Wang.
Thank
And
you.
next, Dr. Alucilia.
Yeah, I just want to express my appreciation for that interesting topic. I've never known about this problem or this business of parrot and pigeon.
Yeah.
Heard about it, but never realized that it's a big research area in Taiwan. So you mentioned that you are in Tainan.
I graduated from NCKU in Tainan, but now the NPUSD is located in Pingtung, the southern part in Taiwan. Yeah.
Okay, because
So maybe
I had
next
lived
time
in Taiwan,
yeah.
in Tainan.
Yeah. So maybe next time, if we have chance, we can invite you to visit NPUSD. Yeah, and to
Sure. I lived in
establish the
Tainan.
collaboration. Yeah.
I
you
lived in Tainan
China?
for 6 months.
Oh, in NCKU?
No, I was a research intern at the Asian Vegetable Research and Development Center.
AVRDC.
Yeah, AVRDC.
And
I
I will
know
be
that.
going there, Banji, for the April trip.
Oh,
I will
it's
be
very,
going to AVRDC as well
okay.
as
Very famous
NTU,
international.
NTU,
Yeah.
SNT.
Yeah, I was
Yeah.
a research intern there. That's where
Oh, okay.
that's where I learned my
So do I.
experiments. That's where I
Oh,
learned
okay.
my field experiments.
Yeah. Very good. Very good. Yeah. So we all have the connection with Taiwan, right?
Right.
Yes, Banji, maybe
Yeah.
we go there in August.
That's right.
Yeah.
Have
Yeah.
a reunion there somehow, right?
Yeah.
My first training in AVRDC in the 80s.
Oh.
Yeah.
On
Okay.
pheromone, yeah.
Yeah,
Okay.
I was trained at AVRDC for my first field experiments.
Yes.
Very good. Very good.
Okay.
Yeah.
All right. Thank you.
Thank you.
Thank you.
Okay. So thank you
And-
very much. Oh, sorry, go, ma'am.
Tad, we welcome everybody, invite everybody to become member of GARD.
Yes.
Dr. Kwang, we want to
Yeah.
join our GARD.
Okay. No problem.
Yeah, there you go.
Yeah.
Give your information to Tad. We'll be glad to have you part of GARD.
Okay.
Let's invite the group.
Yeah.
Yes,
Okay.
ma'am. We will. Okay.
Thank you.
Okay.
Well,
So
thank you.
please, Kavaja, Professor Kavaja, can you
Yes.
transfer my email or information to the professor?
Yes. Yes.
Okay.
Yes.
Thank you.
Dr. Kwang. Okay.
Thank you.
All
Thank
right.
you.
Thank you. Okay.
Thank you, Dr.
So-
Kwang.
Yeah.
Okay. So thank you very much, Dr. Kwang, for that presentation. It was actually very interesting, and many of our graduate students right now are very interested with your research as well. So, of course, moving forward to our ceremonies, we would like to actually award you your certificate of appreciation. So, of course, the Global Alliance for Rapid Diagnostics of Michigan State University awards this certificate of appreciation to you, Dr. Kuo Ping Chuang of the National Pingtung University of Science and Technology, for the presentation entitled "The Rapid Diagnostic Kits Development for Companion Parrot and Pigeon Virus," given at the 2023 Global Alliance for Rapid Diagnostics Forum, the role of rapid and innovative diagnostics in the One Health Perspective, held last March 25, 2023, signed Dr. Evangeline Alucilia, and of course, yours truly, Dr. Tadeus M. Carvajal. So let's give a virtual warm of applause to Dr. Kuo-Ping Chuang of National Pingtung University of Science and Technology. Thank you, Dr. Dr. Chuang, and hopefully Dr. Alucilia and Dr. Ameline will be contacting you regarding about their visit within the year. Okay.
Okay. So Professor Carvajal, can I have
Yes.
this certificate file?
Okay. Yes, we will be
Yeah.
sending it through email, Dr.
Okay.
Chuang.
Okay.
All
Thank
right.
you. Thank you. Thank you.
Thank you. All right. So, of course, we have actually a very interesting of 4 lectures in the past 2 hours, and I hope that all of you were motivated in pursuing research and also talking and discussing with our speakers from the East and Southeast Asia region, Southeast Asia region of the Global Alliance for Rapid Diagnostics. Now, to end our program, I would like to call on for our closing remarks, of course, our very own Dr. Divida M. Ameline, the director of the Center for Natural Center for Environmental and Natural Science, okay, research center in De La Salle University. So, ma'am.
Okay, thank you very much, Tads. I don't want to end this forum. I have so much fun and I have so much learning from this forum. I would like to thank. That's why I wouldn't like to end it, because there are so much important information and sharing that has just happened, no? And I'm hoping that we all still get connected even after this forum. So I will make my, I would like to thank first deep appreciation again for the successful forum for the East and Southeast Asia. First, with the mother of GARD, I would like to thank Dr. Evangeline Alucilia for always approving this East and Southeast Asia forum. It's always is a successful forum with also outcome of collaboration, no? Every single year. And also would like to thank Chelsea and also the group of Dr. Alucilia for the 2023 GARD Forum arrangement. I also would like to thank our speakers, all high talk regarding rapid diagnostic, and all of us can relate with that. And me personally, I'm already connected with all of you on how we can connect, no, with regards to what you are doing. So we'd like to thank Dr. Yamazaki, Dr. Destura, Dr. Angeles, and Dr. Kwang. So I'm pretty sure that we will, some of us will be connected with you, no, even after this GARD forum. And lastly, I would like to thank Dr. Tadeus Carvajal for the wonderful arrangement of this forum, and also to his support staff. This will not happen without the help of his support staff as well. So at this point, I would like to officially close the forum, but let us continue to communicate and collaborate in this topic of rapid diagnostic, no? For One Health perspective. So with that, have a wonderful day, everybody, and please enjoy your lunch. Thank you.
Okay, thank you, Dr. Ameline, okay, for this. And of course, we would like to encourage our participants right now who are watching that there would be a women in GARD panel later tonight. Okay, later tonight, that would be Philippine time, but that would be tomorrow morning, U.S. Eastern Standard Time at 8 a.m. And then the mock simulation at 9:30 a.m., which is Eastern Standard Time. So, of course, Dr. Amaline will be part of that panel and the mock simulation that will allow our participants of the GARD conference to join and interact in the situation as it evolves. So hopefully everyone, you can join again Dr. Amaline, okay, and also the women in GARD for this particular session. So of course, thank you very much everyone for all for listening to us and participating. We hope to actually see you next year for the next GARD 2024 forum. This has, of course, we have sponsors coming not only from the GARD but as well as here in the Philippines to help in trying to establish this continued exchange of ideas and discussions from people around the world. Now, thank you very much, everyone, and please look up the schedule for other potential sessions that are coming from different parts of the world. And we hope that this network, please communicate with us. We like to talk to you, and we can actually try to share ideas in coming up with addressing issues in the One Health perspective. Okay, thank you very much, everyone. To Dr. Kwang, to Wataru Sensei, to Dr. Joma Angeles, and as well as Dr. Raul Destura. Okay, so thank you, po. So we will have just a mandatory group picture on this case. So I hope that you can open your cameras. I apologize that I can't. Currently, we are in Ilocos, in the northern part region, and I'm having internet problems at my at where I am right now. Okay, so there. So Adon or Paulo, could you um, facilitate in the group picture? So I'd like to also invite the support staff and thank them as well, such as Miss Frances Paula Miral, Mr. Jawen de la Luna. Of course, we have Mr. Rain Bello, Paulo Ramon Pacheco, Melissa Colo, Miss Eden Joy Alata, and also Mr. Brandon Cyril Lira. Okay, so there. So okay, Paulo, okay, I think Pau, ikaw na lang if ever.
Yes, I can take the picture po. So please turn on your cameras and smile at the webcam. Okay, so 1, 2, 3, smile. Another one. Please hold your smiles again. Thank you. 1, 2, 3. Okay.
All right, thank you very much. Okay. Okay, so thank you very much to all of the speakers. We had a wonderful time, and of course to all of the participants. Please enjoy the rest of the week of the GARD Forum 2023. Okay, thank you very much to everyone. Okay,
Bye,
congratulations.
everyone. Thank you.
Bye-bye.
And once again, from me, thank you so much. And I would like to extend a special invitation to everybody. So,
Okay.
Mr. Kwang, you're already in, okay? You're already a member.
Yeah, I'll be sending the information po, ma'am.
Yeah,
Thank
okay.
you, thank you, thank
Dr.
you.
Yamasaki as well.
everyone.
And Dr.